Thank you very much for a very wonderful talk. And we will have a discussion as the last session.
Now, I would like to invite Thị Ngọc Bích as our second speaker for this session. Bích, the time is yours.
Okay, I will share my screen first. Can you see my screen?
Yes, I can see your screen clearly.
Yeah, thank you very much, Safirina and Maria, to give me this opportunity to be here to talk about the microbiome research in Vietnam. And also in this opportunity, I also want to deliver a little bit about the results in our research in Vietnam also. First of all, I would like to introduce myself. I am Bích from Vietnam with a high burden of infectious disease country. And I now work at Oxford University Clinical Units Research Unit in Vietnam. We focus on the new and re-emerging diseases such as COVID-19, influenza. We also focus on tuberculosis, dengue, central nervous system infection, and zoonosis, particularly in collaboration with the National Hospital for Tropical Disease in Hanoi. We are trying to understand the generation transmission and impacts of antimicrobial resistance in both hospital and community setting in different aspects. Why focus in Vietnam? Because Vietnam is a low and middle income country with high burden of infectious disease and a hotspot of antibiotic resistant genes. The increasing of antibiotic resistance has been recognized as a consequence of overuse and afteruse of antibiotic in both human and animals. In Vietnam, the percentage of the change in total use in human from 2010 to 2020 was 328%. And the total use for animal in 2020 was more than 1,000 tons. The amounts of antibiotics reflect a high pressure of antibiotic on the human gut and animal gut microbiota in Vietnam. In our previous study showed that the gut microbiome of healthy Vietnamese adults and children has been recognized as a major reservoir for antibiotic resistant genes. In fact, we detect about 88% of the feces from healthy people contain NCR1 gene, that's conferring resistance to colistin, and 100% of the sample contains gene conferring resistance to quinolone antibiotic. Also, 77% of the sample contain genes conferring resistance to cephalosporin, which is a very common in the community setting. We also found this gene in feces from animal and water and food collected in Vietnam. And what we know about the microbiota, we know that human gut microbiota is our second brain, and with associated with the prevention of infection and some immune mediated and metabolic disease. The alteration of the human gut microbiota affected by the food what we eat, our living condition, and the drug we take, such as antibiotic. Research in high-income countries have shown that antibiotic may put the host at risk, at increased risk of infection due to the overgrowth of bad bacteria. and also research in high-income countries, so that the human gut microbiota often is rather resilient and recover after a course of antibiotic. The resilience and recover of gut microbiota, like a case of bamboo, the bamboo will pull, we bend in the direction that we pull, but it's able to be the initial after that. So the resilient and robust microbiome can minimize the periods of the microbiome disruption, leading to reduce the risk of infection. There are 4 factors that's related to the microbiota resiliencies after antibiotic exposure, including capability to maintain its functional redundancies, abundance of opportunistic pathogen, and antibiotic resistant gene reservoir. Most of the study were conducted in high-income setting with the low prevalence of antibiotic resistant gene and also less frequency of antibiotic use. But how about the gut microbiota of healthy human in low and middle income country with high level of antibiotic resistant gene and also high frequency of antibiotic use? So we conduct this study to explore the long-term impacts of antibiotic on the diversity of the human gut microbiota in lower and middle income country like Vietnam. And also we evaluate whether repeated exposure to commonly used antibiotic induce the human gut microbiota with high prevalence of antibiotic resistant genes to be resilient to further antibiotic effect. So we conduct the study at our prospective household-based community cohort, which was established since 2007 for assessment the influenza and like influenza infection that's located in the North Vietnam, and this cohort still remain typically agricultural lifestyle with a high burden of infection as our previous study report, and also lower socioeconomic status and poor education. To examine the impacts of antibiotic on the chains of the gut microbiota of the individual in this cohort, we conduct a longitudinal study over a period of 6 months, and we interview the healthy status of the participant and also about the antibiotic use information in the preceding weeks, and monthly we collect the fecal sample of the participant for microbiome profiling. In the cohort, we had 80 households and 265 participants involved in. For this study, for the assessment, the impact of antibiotic use on the chains of the gut microbiota, we select 63 participants from 22 households, and of which 32 participants use antibiotic at least 1 course during 6 months, and 31 participants did not use antibiotic. So the total, we have 373 samples were included in this study, of which we recognize that 57 samples were collected within 4 weeks after exposure to antibiotic. So to compare the microbiodiversity between our study cohort with the cohort with less antibiotic, we conduct a comparison between the our cohort adult with the adult from our cohort and Dutch adult who did not use antibiotic at least 1 year before sampling. In this study, we use 16S RNA sequencing and for identify the bacterial composition, and then we do real-time PCR to identify the antibiotic resistant genes in the feces sample, and also other statistic method to analyze this. So what we found. We did not observe a difference in alpha diversity matching over time at the population level. However, we did observe a very strong perturbation among individuals. When we examine the effects of antibiotic on the alpha diversity, we use the linear mixed effect model, and we found that the ficus carries gene conferring ESBL and carbapenem genes did not affect the alpha diversity. Meanwhile, the use of antibiotic has only an effect on the microbiome richness for very short periods, while the microbiome diversity is less affected. So when and then we color the change of the diversity between the baseline and the subsequent time point. The figure in the left side is the change in the number of species between the first month of the study and the 5 subsequent time points. And the right side is the change in biodiversity as the same manner. The dot spot inside the cell indicate where the time point where participant use antibiotic. So we can see that among individuals who use antibiotic, we observe the diversity reduced at the time point when the participant use antibiotic, but it gradually narrows at the subsequent time point. And in the among individuals who did not use antibiotic, we observe more stable, the diversity more stable over 6 months. So when we look at the impact of antibiotic use on the microbiome community structure and composition, we compare the sample exposed to antibiotic with the sample did not expose to antibiotic. We observe that the microbiota of the cohort stable and was not affected by antibiotic. I mean, the stable of the microbiome community structure did not affected by antibiotic use. We also did not observe any general altered upon the antibiotic use. It means that also the abundance of texture was not disturbed, including bacteria susceptible to cephalosporin. Actually, this finding was as different from previous study in high-income country, which
you have 2 minutes left.
Okay, which show a drastic reduce in biodiversity and the strong effects of antibiotic on the microbiome composition structure in the human gut microbiota. So we examined the microbiota with in relation to the antibiotic resistant gene pattern, and we did not observe strongly perturbated by antibiotic pattern. So we think that there are many more beta-lactam genes has not been assessed, and it needs to comprehensive investing. because the modest of effects of the antibiotic in the is contrast with the previous study. So we hypothesized that the microbiome diversity in our study cohort may have already been reduced by extensive past antibiotic to an extent that the effect to the further perturbation would be limited. So we conduct a comparison between the Vietnamese and Dutch. When we observe that we did not observe any significant difference in the species lineage, but we observe a significant a difference in the biodiversity. It suggests that the reduction in microbiodiversity as a consequence of frequent antibiotic exposure. We also observe that in Vietnam, Enterobacteriaceae family, including Klebsiella and Escherichia caesuala, enriched. Meanwhile, in Dutch Edo, the gut microbiota of Dutch Edo enriched.
Sorry,
by...
Big, but your time is up. Could you please go to the... Okay, thank you.
Okay, in conclusion, in this study we observed a modest reduction of the gut microbiota diversity within less 1 month of the antibiotic use, and the less diversity in the gut microbiota at baseline as compared to Dutch individual from a setting with less antibiotic use. We then hypothesized that frequent or chronic antibiotic exposure may put the microbiota to a different steady state that is less diverse but more resilient to disruption by antibiotic use. So thank you very much for giving me this opportunity to talk here. Also, thank you to our unit, and thank you for your listening.
Thank you, Big.
Yeah.
I will give back to you for chairing the next speakers.